Now, another important layer of the ground tissue system or another important layer in fundamental tissue system is called as pericycle. Now, where do we see the pericycle? For example, this is endodermis. As we studied before, this is endodermis, and the layer inside the endodermis will be present. This layer is called as pericycle. This will be vascular bundles. Okay, the layer between the endodermis and the stele is called as, the layer in between the endodermis and the stele is called as the pericycle. Between endodermis and stellar part or stele is known as pericycle. It is formed in the it is a layer, it is with one or more layer cells in thickness. It may be in one layer in thickness, or you may have it one or two layers of thickness present in between the endodermis and the pericycle. It acts as a boundary to the stellar region, like it protects the stele from the endodermis and the cortex tissue outside. So, it can be called as the boundary line to stellar part or stele. Now, this is also called as outer layer to the intrastellar region. No, we will study about this is about normal endodermis. It is a barrier between endodermis and the stellar part. It is also called as boundary to the stellar region, and it is present to the outer layer of the intrastellar region. Now, here we will talk about in what is the arrangement of or what is the type of cells present in the roots, stems of the root. If you see the TS of root, what is it, a sclerenchymatous pericycle or parenchymatous? In case of stem, whether it is sclerenchymatous or parenchymatous, or it has some special type of tissues. Okay. Now, we will talk about roots. In roots, all these are important. In roots, the pericycle is parenchymatous. While in cucurbita and aristolochia, this is very important. In cucurbita and aristolochia, the pericycle is sclerenchymatous. And in case of helianthus, sunflower, in case of helianthus stem, you will see the alternate bands of both parenchymatous and sclerenchymatous tissue. You will see the alternate bands of both parenchyma and sclerenchyma. These 3 points are very important. In roots, the pericycle is parenchymatous. In case of cucurbita and aristolochia, sclerenchymatous tissue is made up of pericycle is made up of sclerenchymatous tissue. In helianthus stem, you will see the alternate bands of both the parenchyma and sclerenchymatous tissue present. Sometimes this pericycle also consists of sclerids, and sometimes pericycle also consists of sclerites and fibers. As I said, pericycle, you also see the presence of sclerites and fibers. So, how these sclerites and fibers are formed? These are formed as a continuous layer. Now, what is the example in this type? If you see the presence of sclerites and fibers in pericycle in a form of a continuous layer, then this type can be taken can be seen in an example like aristolochia. Okay. Now, in case of roots of angiosperms, the pericycle is uniseriate. All these points are important, and except in, all these points will come for your entrance, uniseriate except in hydrophytes and parasites. What does it mean? Roots of all angiosperms are uniseriate except in hydrophytes and parasites. Means in hydrophytes and parasites you are not seeing the arrangement of pericycle in uniseriate form. You may see the variation in that. We said earlier that in roots of angiosperm the pericycle is uniseriate form except in some hydrophytes and parasites. Okay. Now in case of monocots like smilax, agave, palms, and in some dicots like morus, you will see the presence of pericycle in a multiseriate manner. And in case of, for example, if we take the example of gymnosperms, so what will be the arrangement of pericycle? If we see the arrangement of pericycle in gymnosperms, you will see that pericycles are in multiseriate order. Okay. So here when we conclude this pericycle is nothing but a barrier between the stele and the endodermis. It is the outer part of the intrastellar region. It acts as a boundary between the endodermis and the stellar part, and it is made up of parenchymatous cells. In cucurbita it is made up of sclerenchyma, and in case of Aristolachia, and cucurbita it is made up of sclerenchyma, but in some cases it is made up of alternate bands of parenchyma and sclerenchymatous cells. Now in some cases it is uniseriate, and in roots of angiosperm you will see the presence of uniseriate, and in hydrophytes and parasites it is not uniseriate. In monocots like smilax, palms, and dicots like morus you will see the multiseriate arrangement of pericycle, and in gymnosperms also you will see the arrangement of pericycle in the form of multiseriate forms. Now we will talk about the functions of pericycle. Why we are in need of studying pericycle in detail is, like as an anatomist or studying the histology, before going to the anatomy of internal structure of any TS of section, we should know in detail each and every section of fundamental tissue system and vascular tissue system. So we can't just say pericycle one layer or 2 to 3 layers. We need to know every function of pericycle. So what are the functions of pericycle? Okay, the major function of this is, this can be considered as an origin for all the lateral roots. It will be acting as an origin of lateral roots. Now in dicot root, at the time of secondary growth, this pericycle get converted into phellogen. In the plant parts where you will see the presence of anomalous secondary growth, we will also see the presence of secondary cambial rings. So these secondary cambial rings are also originated or formed from the pericycle. So what I was telling is, at the in plants where there is anomalous secondary growth, we will see the occurrence of secondary cambial rings. These are formed from pericycle. Okay. And here one more is, when we were talking about the characteristics of pericycle, we said that some of the pericycle layer is made up of sclerenchymatous. In some plants you have said that it is made up of bands of parenchyma and sclerenchyma. If we say that pericycle is made up of a layer of sclerenchymatous cells, so automatically we can say that this sclerenchymatous pericycle will give mechanical strength to the plant. Gives mechanical strength. Now again, this pericycle is also useful for the storage of food material. This food material may be in the form of starch, as we said when we are studying about the general characters of pericycle. These are the functions. In addition to that, we can also see some type of secretory tissue, laticiferous tissues are also seen in the some secretory tissue and latissiferous tissue can also be seen in the pericycle. So basically pericycle is the basic origin place where the origin of lateral roots occur. In dicot root, in the time of secondary growth, they get converted into phyllogen, and in some plant parts where there is a occurrence of anomalous secondary growth, you can also see the occurrence of secondary cambial rings. So how does these secondary cambial rings form? This may be formed with the help of pericycle because of the presence of pericycle, and some pericycle have sclerenchymatous cells in them. So those pericycle with sclerenchymatous tissue will give mechanical strength to the plant body, and this pericycle is also useful for the storage of food material. So this is about pericycle.